Age-related symptoms such as urinary retention and poor urinary stream have been attributed to detrusor underactivity. Previous studies showed that urethral relaxation during bladder contraction was diminished in aged rats. Nevertheless, L-arginine couldn't restore diminished urethral relaxation. Thus, we hypothesized that age-associated oxidative stress would impair NO-STC system, which induces urethral relaxation. We tested this hypothesis by measuring non-oxidative stress marker in the bladder, and also simultaneous recording intravesical pressure and UPP using a novel NO-independent stimulator of STC. We used female rats. We evaluated urethral activity by simultaneous recording of intravesical pressure and UPP under wartime anesthesia. We administered the drugs after the bladder contraction had stabilized. We compared bladder and urethral pressure parameter between young and aged rats. In addition, we averaged and compared these parameters before and after drug administration, respectively. We performed immunostaining using the bladder of rats. We used 8-OHDT and MDA antibody as a primary antibodies. These results were evaluated by several persons and scored on 3-point scale. Results of young and aged rats. Baseline UPP and UPP changes were significantly lower in aged rats than in young rats. These results mean that aged rats have disorder of NO-dependent urethral smooth muscle relaxation. Results of young rats. After administration of SNP and BAY, baseline UPP and UPP nadir were significantly decreased in young rats. These results mean that these substances relaxed urethral smooth muscle. After administration of L-arginine as an inhibitor of NO synthase, the baseline UPP was significantly increased, but UPP change was still lower compared with control in young rats. These results mean L-arginine couldn't restore SNP and BAY-dependent urethral smooth muscle relaxation in young rats. Results of aged rats. After administration of SNP and BAY, aged rats responded similarly to young rats. However, after administration of L-arginine, baseline UPP as well as UPP nadir were significantly increased. UPP change was not significantly different from control. And this means that L-arginine could restore SNP and BAY-dependent urethral smooth muscle relaxation in aged rats, possibly due to decreased NO-STC system in aged rats compared with young rats. As shown in the image, 8-OHDG and MDA levels were higher in aged rats than those in young rats. As shown in this graph, aged rats had significantly higher staining. UPP change during urethral relaxation was significantly smaller in aged rats than in young rats, as well as recent to our study. Aging is assumed to induce dysfunction in urethral smooth muscle relaxation during boiling, possibly due to the decrease in NO synthesis by age. Because NO-independent stimulator could restore impaired urethral relaxation in aged rats, age-associated oxidative stress in the NO-STC system might be involved in the impairment in bladder and urethral coordination. Conclusion. Thank you for listening.